So in this presentation, we want to look at enzyme regulation. What is very important about this topic here, or this subtopic of this concept, is that you don't want enzymes to work endlessly without stopping. So what mechanisms do we have in the body that makes sure that there is a stop to enzymatic action? Okay, or if we can't say that, what do we have in the body that makes sure that the rate of enzymatic activity slows down or basically increases? So we have concepts to do with these mechanisms that we want to look at. So we start with this scenario. We are having an enzyme. This enzyme is actually very important in genetically modified maize. So what this enzyme does is basically regulating nitrogen metabolism. We know that nitrogen is actually very important in plants. So what this enzyme called glutamine synthase does is to regulate this metabolism, this nitrogen, in this genetically modified maize. So scientists have actually noted that this enzyme is actually very, very active when there is deficiency of nitrogen. What the scientists also discovered was that there are other small molecules in this maize that bind to this enzyme and change its shape, and then making its enzymatic activity faster. So enhances enzymatic activity. So in a separate experiment also, you have scientists also discovering that when you have glutamine, when you have it in large amounts, it basically reduces the activity of this very enzyme, glutamine synthase. Okay, so even when you have high substrate levels, so the presence of glutamine makes the enzyme not to work very well. Okay, but the enzyme works very well when nitrogen is actually deficient, in nitrogen deficient conditions. Okay, so the things that we have talked about actually rotate around issues to do with enhancement of enzymatic action or a decrease in enzymatic action. These are the things, and I think you can now categorize this. Now, where do we have this enzyme working very fast, and where do we have it being inactive? So you have those two things or two conditions. So that means that we are talking about regulation of this enzyme. Under what conditions should we have it working efficiently, and under what conditions do we have it working less efficiently? And that's why we're talking about this regulation. So one of the regulation types that we shall talk about, and that we actually need to know in this presentation, is what we call allosteric regulation. So allosteric regulation is what we need to know and how it basically happens. And then the second one is covalent modification. Okay, covalent modification. What is it? We also need to understand that. Okay, the other thing that we really need to know is an issue to do with feedback mechanism. So when we talk about feedback mechanism in regulation of enzymes, what is it? Can we explain it? And why is it very important in maintaining balance? Then lastly, we need to address issues to do with post-translational enzyme regulation. What advantages do we have in this concept in relation to regulation of enzymatic action at the gene level? Because we shall also look at that gene level regulation of these enzymes. Okay, so that is basically it. Still, this question again points to real application of enzymes in at industrial level. Okay, and in this case, we are basically looking at the application of enzymes in getting what we term as GMOs, genetically modified organisms. And in this case, we're talking about genetically modified maize. Okay, so we don't want to have metabolic activities happening endlessly without control. We need to produce products that are in sufficient amounts for efficient metabolic activity. If we have a situation where the products are produced in large amounts, the body may not be able to handle. So there needs to be a balance between the products and the substrates. So if we have a lot of substrates, and then it means that the products will be less. So we want to have a balance that it is from the substrate that we get the products, but we should not have a lot of products depleting all the substrate. There should be that balance for homeostatic states, or what you call to have that balance, okay, between substrates and products. And of course, the regulator of these reactions are actually enzymes in between, because we have enzymes that will make you get products, or you have enzymes that will speed up the reactions that give you back the substrates. Okay, so which that definitely means that the enzyme, if you can regulate how much enzymes you have in between, it means that you can regulate how much products you will have this side and how much substrates you'll be left with on this side. So that's why enzymatic regulation is actually very important. And we're going to look at this mechanism. The first mechanism is what we call end product inhibition. Okay, so the meaning of this is that if you form more of the product this side, this product will act on the enzyme such that it makes it less efficient, such that you don't have more products being formed. So it is this product acting on the enzyme such that you have less of the product being produced. So that is end product inhibition. It's what you can definitely call a negative feedback mechanism, okay, which is very important in homeostatic mechanisms. So you have an enzyme that is inhibited by the product that it forms. So the second way is by having enzymes being synthesized in inactive forms and only converted into active forms when the conditions are actually ripe. Okay, so that means that these enzymes are only active when they are needed. That is very important for controlling metabolic reactions, because in the end, if you produce, for example, enzymes that are active in the immediate environment, it can actually cause this damage or injury to tissues. So, for example, you have trypsin. Trypsin is the active form of trypsinogen. So the trypsinogen is secreted in its inactive form and has to be converted into trypsin for trypsin to have its action. So you could imagine if it is produced immediately as trypsin, it could create a lot of digestive activity in its locality before being turned into if it happens that it doesn't need to be converted into the active forms. Okay, so we have the third one, which is genetic control. Every molecule that is synthesized in the body, or that is actually made in the body, that molecule has to be synthesized based on the information that is received from the nucleus. And for the case of proteins, and in this case we're talking about enzymes, which are basically proteins, the synthesis of these proteins, the information has to come from the DNA. Okay, because we know that DNA and then RNA, messenger RNA, transfer RNA, and ribosomal RNA. These are very important in the synthesis of proteins. Okay, so that means that if you control the synthesis of these proteins, basically you're controlling the enzymes that you're having. So that means that you either want to have more of the enzymes or you don't want to have a lot of these enzymes. Okay, so at the gene level, it's possible to control enzymatic activity. Okay, the synthesis of these enzymes follows a signal, or follows signals, or what you can term as cell-to-cell communication mechanisms. So cells do communicate that, okay, this is actually needed, this is what needs to be synthesized. And these are basically about cell signals or cellular signals that inform the cell, especially the nucleus, what kind of proteins or molecules need to be synthesized. Okay? So they can be hormonal signals or basically cellular signals. And then we have what you term as allosteric regulation, where you have another molecule binding to another side apart from the active side and then causing a transformation of the active site. And this consequently results into the enzyme not being able to carry out its function because the active site has been altered. So you can have a number of those molecules that naturally exist within the body to basically carry out that function. And then we have covalent modifications. Here, you're having the enzyme not being able to carry out its function just because the primary structure of this enzyme has been altered, because you have attachments to the enzyme that these attachments are through the covalent bonds, and that's why we are actually terming this as covalent modifications of the enzyme. Okay, and this attachment that creates this covalent bonds actually alters the shape of the enzyme and making it inactive. Okay, so basically it cannot function quite well, and as a result of this interaction can be reversible or irreversible depending on what the body may want.